Thanks everyone for coming. So for today, I'll be the first speaker, and the title of my talk is Sex-Specific Mechanisms of Brain Aging, and I'm an associate professor at Brown University. So I will share my screen. How does that look? Good, thank you for the thumbs up. All right, so just a couple quick disclosures before I start. All right, so in my lab, we study the molecular mechanisms of brain aging and neurodegeneration. I think as many of you already know, aging is accompanied by a loss of cognitive function, and that includes decline in learning and memory. So this is very common as we age. And at the same time, aging is the greatest risk factor for a number of diseases, and that includes diseases of the brain, so neurodegenerative diseases such as Alzheimer's disease. So for example, before the age of about 65 or so, pretty much nobody gets Alzheimer's disease. But after that age, there's an increasing prevalence of Alzheimer's disease as well as other neurodegenerative diseases. So despite these well-known issues in aging, we really don't have a handle on the molecular mechanisms underlying brain aging and neurodegeneration. And so this is what my lab is interested in studying, with the idea is that if we can understand these mechanisms, then hopefully we can come up with ways to intervene and improve healthy aging of the brain. Now, a lot of the work that's been done, for example, in the context of Alzheimer's disease, has been looking at a part of the brain known as the hippocampus. And so this is a cross-section through a mouse brain. And the hippocampus, which is the center for learning and memory functions, is right up here in the mouse. But there's another region of the brain that we have become very interested in recently. We feel this is an area of the brain that it's fairly understudied in the context of aging, and that's the hypothalamus. And so the hypothalamus has a number of diverse functions that are really critical for healthy aging. Many of those functions are listed here. And so all of these things tend to change with age, right? So we often undergo weight changes with age. The hypothalamus regulates body temperature. It's harder to regulate your body temperature with age. Hypothalamus is also critical in pituitary control, so it's the H in the HPA axis, as well as these other functions that are known to change with age. Lastly, at the bottom here, sleep-wake cycles, so our circadian rhythms and sleep in general, definitely altered with age. I think we all feel that as jet lag gets worse and worse with age. But again, really the mechanisms underlying these changes aren't well understood. And so just to give you an idea of where we are in the brain, this is a human brain here, and the hypothalamus is the area that's shown in blue. So it's basically like right over the roof of your mouth. And then in the mouse, where we're studying the hypothalamus, and the data that I'll show today are from the hypothalamus in the mouse, the region is circled down here. So it's down below the hippocampus. All right, so not only do all of the processes that the hippocampus modulates change with age, it's also true that there is more direct evidence that the hypothalamus itself can actually modulate lifespan. So these are just a few studies that have come out in the last decade or so. There's work from Dongsheng Kai's lab demonstrating this. So if you alter the NF-kappa B axis in cells within the hypothalamus, that can actually extend lifespan. There's been a lot of work from Shinamai's group at WashU, also implicating the hypothalamus and lifespan itself. So this is just one of the figures from the paper where they generated the mice known as the Brasto mice. And so these are mice that have increased sirtuin and SIRT1 in particular activity in a couple of regions of the hypothalamus. And those mice have an extension in their lifespans. And then down below, lastly, there's a whole body of research from Andre Bartkey's lab implicating the growth hormone axis, including neurons in the hypothalamus, in aging and longevity. And so this is just one particular knockout mouse with an alteration in the hypothalamus. And these mice actually you can see a pretty significant increase in lifespan compared to the normal controls in blue. Okay, so hypothalamic functions change with age. The hypothalamus itself seems to be modulating aging and healthy longevity in some way, yet the molecular mechanisms responsible are not very well defined at this point. And so this is a question that two of my graduate students wanted to take on starting a few years ago. It's really a new area in my lab. So these are both brave graduate students, Caitlin Hadanovic, who's a neuroscience graduate student here at Brown, and then Dodo Yu, who's an MCB graduate student at Brown. They're both Carney Fellows, so they're both fellows of the Neuroscience Institute here at Brown, and Dodo is also a Data Science Master's Fellow. All right, so what we wanted to do to begin was just really sort of a very simple experiment. So we simply asked, okay, how does gene expression change in the hypothalamus at the single cell level? So we wanted to look across all of the different cell types in the hypothalamus, and we compared young mice and aged mice at 3 and 24 months. We basically just dissected out the whole hypothalamus in these mammals, and then we isolated nuclei for sequencing. We had to use nuclei in this case because in the case of the brain, if you dissociate and try to take out and retrieve single cells, most of the neurons will not survive that process because the neurons are so fragile. So instead we sequenced RNA from the nuclei. And we sequenced about 40,000 nuclei using the 10x platform. And this is just a UMAP, kind of give you an overall view of what these data look like. So on these UMAPs, I think most people are familiar, but remember that every dot represents a cell, or in our case, a nucleus. And we were able to obtain large numbers of neurons in our experiments, so those are shown in green, as well as the other major cell types in the hypothalamus. So for example, we have astrocytes, oligodendrocytes, microglia, even tanycytes, which are rare cells in the hypothalamus. So we felt pretty good about our data set. And without getting into too much detail, within just the neurons, we were able to identify 35 different subtypes of neurons. And those are shown here in this dendrogram. And we could identify, for the most part, the identity of these different types of neurons, as well as a major neuropeptide that's secreted from them in the cases where there's a major neuropeptide that's generated from the cell type. We also took advantage of spatial data that was available through the Allen Brain Atlas and projected our data sets onto their spatial data. And this was really just sort of a validation to validate the cell types that we pulled out. So for example, the astrocytes are shown here, and they're pretty much distributed throughout the hypothalamus. Whereas in the case of specific types of neurons, they were more localized within the hypothalamus, which is what we expected. And so just highlight a couple of examples here. CAR-T neurons are up here, and then the POMC neurons, which are found in the arcuate nucleus, they're feeding neurons. They are these bright yellow neurons down here at the base of the hypothalamus. So we felt pretty confident about the identities of our neurons that we were able to find in the hypothalamus. All right, so the big question that we had is what happens with age? So a few sort of points to give you an idea of the quality of the data set. The first key point is that we did not see major changes in the composition of the hypothalamus in our data sets with age. So this is just a UMAP where I've colored the cells either blue or red for young and aged. And you can see that each major cluster in the UMAP contains both ages, and so we didn't derive completely new clusters from the aging data set, and this was sort of a good thing, because it is known that the brain doesn't change majorly in composition with age, at least not with non-pathological aging. There isn't a lot of cellular death with age, and so it gave us good confidence that there wasn't something technically wrong with our data set, particularly with age. We were able to get back the cell types that we expected. And on the right, this is just another way of depicting this quality control measure. We had 4, I didn't mention this, but we did 4 replicates per age, so 4 young and 4 aged animals. All right, so importantly, we did pick up cell type specific changes in gene expression with age. I think this is generally sort of expected. So in this strip plot here, every dot now represents a gene, and any dot that's in color is representing a statistically significant change with age. So for example, astrocytes here had lots of changes with age. The neurons are down here in green, also lots of changes, but different from the astrocytes. And then there were other cell types that didn't seem to have as many changes with age, though in a lot of cases we didn't have a lot of cells for that cell type, so it was a little bit harder to pick up significant changes with age due to low cell numbers. All right, then just a little bit about the types of pathways that are altered with age, and these were very cell type specific in nature. So for example, in the astrocytes, we saw a lot of changes in cholesterol homeostasis. We also saw alterations in mTOR signaling genes, and those changes in the TOR pathway genes were also found in the neurons, but then neurons also had other distinct changes with age, distinct from other cell types. And microglia, for example, gave us lots of changes in immune pathways and inflammatory pathways. So somewhat expected, but new insight, I think, into how these different cell types are altered with age. Now, just zooming in on the neurons were quite interesting. So I took off, just for simplicity, I took off the names of the different neuronal subtypes, but those 35 clusters that I showed earlier, those are on the left side over here. So each row is a different type of neuron. And then I've also taken off the names of the molecular processes that were altered with age, just for simplicity. But this is a gene set enrichment analysis. And so a few points here. First of all, we found that the different types of neurons were aging quite differently, and some of the neuron types had lots and lots of changes with age across many different molecular processes, whereas other types of neurons had relatively few changes with age, possibly indicating that they're more resilient to the aging process. And then the second point is that when we look at the molecular processes, there are some processes that are changed across almost every type of neuron, whereas other molecular processes seem to be sort of specific to one type of neuron. And we looked a little bit more deeply into the processes that are altered across many different cell types. What we found was pretty interesting, I think, which was that these hallmarks and pathways tend to be neurodegeneration associated pathways. So this suggests that potentially the gene expression changes that we picked up with age here are the gene expression changes that make neurons susceptible to neurodegenerative diseases. So we're very interested in digging deeper into these sort of common mechanisms of neuronal aging, as well as the more resilient mechanisms. All right, so we wanted to ask whether we could observe trajectories of aging across these different cell types in the hypothalamus. So we ran some pseudotime analyses across our different types of cells in the hypothalamus. And interestingly, in most cases, we couldn't define a specific trajectory across any particular subtype. The exception of this was the macrophages and the microglia. And the microglia gave us a very interesting age-associated trajectory. So just briefly, the way that this works, these pseudotime methods were developed mostly for developmental data sets. And so, for example, here, if you have a developmental data set or even an aging data set that represents physical time, that's your starting point. And then if you take away all of the aging information or time information, and then just look at the transcriptome information across each cell, that's what's shown here in B. So this is sort of a loss of temporal information that's been removed. You can apply these algorithms, these pseudotime algorithms, that will then reorder the cells across time. And that new time is known as pseudotime because it's learned from the data set. It's not real time. But in general, what the algorithm is able to do is reorder the cell in a way that represents physical time. And then once you have this pseudotime in place, you can then dig in and ask, okay, what are the particular gene changes that are changing across pseudotime and that are representing aging in our case of the cell. All right, so when we ran this monocle analysis on the microglia in our data set, we got a very nice aging trajectory. And so this is the aging trajectory here. On the bottom, this is the representation of pseudotime. And so young pseudotime is on the left in purple, and then moving across to the yellow, this is going across pseudotime into aging. And then when we project the real-time information back on that pseudotime that we obtained from the algorithm, you can see that the young cells, interestingly, they cluster early in pseudotime, and then lots of aged cells are found later in pseudotime. So it does seem to represent an aging trajectory. But I will also point out that something that we didn't expect to see in this analysis was that some of the aged cells are actually still found earlier in pseudotime, suggesting that at least some of the microglia are maintaining younger features of a microglial state, of a more youthful microglial state, even in the aged brain. So we're very interested in digging into the differences between the young and the aged microglia within the old brain to see if we can understand better how to take those old microglia and bring them back to a more youthful state. And so we've dug a little bit deeper into these signatures across pseudotime. And one thing we did was we took some signatures that were developed by Ido Amit's lab. And these are signatures of either healthy homeostatic microglia, what's known as stage 1 DAM microglia. So these are basically aging microglia, or a third type of microglia. And these are microglia that are undergoing neurodegeneration. So for example, isolated from Alzheimer's brains. So we basically asked, does our pseudotime data set correlate with any of these different signatures of microglial states? And what we found, I think, was pretty interesting. So what we found is that across pseudotime, so again, this is our pseudotime data here, we see the peak of homeostatic microglia pretty early and into the mid stages of pseudotime, whereas homeostatic microglial signatures are gone later in pseudotime. Whereas the age-associated microglia, these DAM1 microglia, these are low early in pseudotime, and then they track upwards across pseudotime, so they become more highly represented later in pseudotime. And then we never really saw much enrichment for DAM2 microglial signatures in our data set. And this actually makes some sense because the DAM2 microglial signatures, remember, are representing a disease state. And our mice were just undergoing healthy aging, not neurodegeneration. We could then ask what specific genes were represented across pseudotime in our data set. And so we could divide pseudotime into 4 distinct modules, which is shown here in the heat map on the left. So module 1 is represented early in pseudotime, followed by genes that come up in module 2, and then 3, and then finally module 4 represents late pseudotime. And I realize that these, the text on this slide here on the right is a little bit small, but I'll just make a couple of general points. As we look across the modules and across pseudotime, initially we see signatures represented that are more representative homeostatic signatures, such as gene regulation and cell signaling, sort of normal functions of the microglia. But then as the cells progress through age, they then have altered signatures that are related to metabolism and organelle homeostasis, followed by an induction of cellular stress, and then finally alterations in quality control, trafficking, and viral processing. And I'll just point out the very last signature that's shown here is regulation of amyloid beta clearance. So it does seem that as these cells get really, really late in pseudotime, they are starting to represent more of a neurodegenerative state. Okay, so a couple of points about the extent to which these signatures that we pulled out are shared across brain regions. So here we're really focused on the hypothalamus, but as I mentioned, there's been a lot of work on the hippocampus, which degenerates very significantly in diseases such as Alzheimer's disease. And so we wanted to ask whether our signatures in the hypothalamus were shared in that brain region as well. And we were fortunate that Nicola Neretti's lab, who's also here at Brown, had done a very similar experiment where they did a single nucleus sequencing of the hippocampus using the same platform. So we basically took our data set and correlated it with theirs. And so we started with the glial cells in the brain. And what we found was that all of the different types of glia that we pulled out in the data set, so oligodendrocytes, microglia, as well as astrocytes, these tended to be correlated across the different brain regions. So at least at the transcriptional level, these cell types seem to have similar signatures of aging. But what was interesting was when we looked at the neurons, we saw very different signatures of aging, suggesting that the neurons in the hippocampus are aging in a very different manner from the neurons in the hypothalamus. And this is potentially very interesting in the sense that it could give us some insight into why hippocampal neurons are more susceptible to neurodegenerative disease compared to hypothalamic neurons. So we're digging into this a little bit more in the future. All right, so in the last few minutes, I want to tell you about a feature of hypothalamic aging that we pulled out that was a little bit of a surprise in our data set. And what I didn't tell you at the beginning was that the way that we set this experiment up was actually only using female mice. And so we think it's really important to understand both male aging as well as female aging. And at this point in time, when we started the these experiments, we thought, okay, we need to just focus on one. We couldn't do both sexes. So we started with female mice. And the reasons that we want to look at both female mice as well as differences between males and females are several. So first of all, it's well known now that lifespan is sexually dimorphic in most species. So in humans, females tend to live longer, and that's true in a lot of mammals, but across other species, there are other cases. So in some species, males actually tend to live longer. And in addition to that, we know that some diseases disproportionately affect one sex. So for example, heart disease is more prevalent in men. In women, we actually get more Alzheimer's disease. That's shown here in this diagram, right? So about 2/3 of people who have Alzheimer's disease right now are women. We don't understand the molecular basis for that at all. There are few studies that actually address the mechanisms of female aging at this point. And so we felt that this was a good time to start doing that and to also start comparing males and females. And then lastly, many interventions that extend lifespan affect only females, right? And so, so we don't understand these differences, again, between why females sometimes respond better to certain interventions and why males respond better to others. But we need to start sorting this out, in my view. Okay, so we just looked at females in this particular study, and that led us to a very unexpected finding. So when we looked at gene expression changes just in the neurons with age, we found that the top most upregulated gene was a gene known as Xist. And this is a gene that is expressed on the X chromosome, and it's a key modulator and sort of the master regulator of X chromosome inactivation. And this wasn't just true in a few different types of neurons. This was pretty uniformly true across all of our different types of neurons. So this is back to those 35 different types of neurons in the hypothalamus that we found. And the Xist is labeled where it's differentially expressed in the different types of neurons. We also found upregulation of a related gene known as Psi-X, shown here, which is also part of the X chromosome, which is known as the chromosome inactivation center. All right, so just as a reminder, in terms of how the biology works in humans, so males are XY, females are XX, and in order to achieve dosage compensation, females express this Xist RNA just from one of the X chromosomes, and that happens during development. So once the Xist RNA comes up, it actually coats just that X chromosome that it's expressed from, and then it recruits epigenetic modifiers that then silence the X chromosome. And so once that X chromosome is transcriptionally silenced, it's actually sort of set aside in the periphery of the nucleus, as shown down here, and then it remains transcriptionally inactive for the most part throughout life. So this is the process of X inactivation. Again, this happens early in development, and it's thought to be stable across lifespan once it occurs. So this was sort of strange that we pulled out this modulator of X chromosome inactivation that's a developmental modulator. So the first thing that we wanted to do was just validate this finding. And we did that in a few different ways. One thing we did was we went in and we did RNA scope for Xist on the hypothalamus, and we looked across a number of different animals, and we found that indeed, when we used this method, there was an upregulation of Xist in the hypothalamus. We also checked by RT-qPCR. Here we're looking at a few different brain regions from the female brain, and we actually saw a bit of a trend in every brain region, though in this case we saw the strongest upregulation in the hypothalamus. Now, importantly, we did not see major changes in Xist in the male hypothalamus, and so that's an important finding because it suggests that this may actually be a female-specific phenomenon. So this is actually data down here on the right. These lines, there was just no detection. We also were very curious to know whether this might be happening in the human brain. It was a little bit difficult to find studies that included females, but in the case of Alzheimer's studies, there were a lot of female data sets out there, in part, I think, because there are more females who get Alzheimer's. And so we looked at a couple different data sets that were publicly available. They looked at two different regions of the brain. And when we specifically looked at Xist expression in these different data sets, we found that indeed in the entorhinal cortex, there was an upregulation of Xist in Alzheimer's patients post-mortem. And this is an important area of the brain that projects into the hippocampus, and it's actually an early area of the brain that degenerates in Alzheimer's disease. We did not see this change in the prefrontal cortex, and this is an area of the brain that is affected in Alzheimer's disease, but a little bit later in the disease. So these are some initial findings that indicate that possibly, at least in the context of neurodegeneration, there are changes in Xist levels. So this biology, something might be going on with X chromosome biology in females as well in age-associated diseases. Okay, so this led us to question whether Xist might be a biomarker of aging. So to answer this question, we turned to a machine learning approach, and we moved back to our own data set, and we just took all of the genes on the X chromosome as input, and we looked only in the neurons because this is where we saw this change most strongly. And we fed those data into a few different actually 8 different machine learning algorithms, and asked if we could identify algorithms that could then predict the age of neurons. So could we correctly identify a neuron as either a young, healthy neuron or an aged neuron just using genes and features on the X chromosome? So this is just the run through of the 8 different machine learning algorithms that we tested. XGBoost tended to be the best performer on our data set. And so we ran XGBoost using a number of trials on our test data set. And we found that about 85% of the time we could correctly predict an aged neuron as aged, and about 67% of the time we could predict a young neuron as young. So for machine learning, that's pretty good performance. We were pretty happy with that prediction. We then dug into sort of looking under the hood and this machine learning algorithm, what's driving the algorithm and allowing it to predict the age of a cell. And so the way to do that is to basically go in and start removing features one by one and ask whether the performance of the algorithm is compromised. So does the algorithm break when you start taking out certain genes? And so that's what's shown here. So this is the performance after removal of these different features. And interestingly, the feature that seems to be most important for this to run is Xist expression. So if we took out Xist, then we lost performance in the XGBoost algorithm. There were other features that also contributed as well. So what we really want to know, of course, is what are the functional consequences of elevated Xist with age? Why is this happening? Is this good for the cell? Is this bad for the cell? Is this just neutral? And I don't have an answer to that today, but that's sort of where we're going with this in the future. Okay, so just to sum up and give you an idea of some of our future directions, today I told you that we have generated basically an atlas of the aging hypothalamus, and we see distinct transcriptional signatures of aging in different cell types and lots of distinct differences between different types of neurons. We also found a female-specific feature of neuronal aging, and that's elevated expression of the X inactivation center with age. And we also find this feature in the Alzheimer's disease female brain compared to age-matched controls. We were then able to apply a machine learning algorithm and show that Xist can be a predictor of neuronal age in our data set. So again, we have a number of outstanding questions here. So what are the mechanisms that are underlying Xist dysregulation in neurons? Does this actually impact cellular age when Xist is changed? We're also very interested in understanding whether there are sex-specific epigenetic changes on sex chromosomes with age. So far we've only looked at females. What's happening in males? Males also have an X chromosome. And then lastly, we're very curious to look into whether the hypothalamus is affected in Alzheimer's disease, because many of the functions of the hypothalamus, such as regulation of sleep and circadian rhythms, are well known to be affected in Alzheimer's disease as well. Yet we know almost nothing about how this part of the brain changes in neurodegeneration. All right, so I will stop there and I can take questions. I'll just acknowledge my wonderful lab here at Brown. And I already mentioned the main drivers of this project, Caitlin Hadanovic and Dodo Yu, with a lot of help also from Lexi Hassel, my research assistant. And I will also acknowledge funding for this work, including a new NSF center to look at sex differences in aging. And I'm happy to take questions. All right, so I will stop sharing. Vera, you can unmute, go ahead.
Okay, well, amazing story, Ashley. I'm just very curious of the continuation. So a couple of questions. So what actually happens to X chromosome? Does it, is it more activated or more silenced with aging?
Great question. So what we think is happening, and this is a lot of hand waving at this point. It's still a model, but what we think is happening is the X chromosome genes overall are elevated with age, and what Xist is doing is it may be coming up to try to get the chromosome back to homeostasis, to try to bring the level of the genes back down. But that's really just the correlative nature that we can, or the correlative data that we have right now. It's not functional data yet. So we're working on, for example, designing CRISPR systems to alter Xist activation itself in an acute way to see if we can actually get at the mechanism a little bit better.
And one more quick question. So I saw one of the changes that you noted was in aged neurons was viral processing. So I wonder if you can comment on that, or if it has anything to do with our beloved transport.
Yeah, no, it's a great question. We haven't done anything to look more specifically at that, but I know your work and the work of John Sedeby here at Brown. I think there's a good chance that there's something going on there, but we haven't looked in detail. Yeah, thanks. All right, Raul, I see you have your hand up as well.
Yes, a fantastic story. Actually, I have also a couple of questions. The first, related to Xist also, that the RNA scope has enough resolution that you could see whether it's expressed on the second copy or it just increased expression on the inactive X.
Yeah, also a really good question that we don't have the answer to. And the reason we don't know is because this was done on inbred mice. And so we can't tell the difference in our transcriptome between the active and the inactive X. And we didn't know we were going to find this. So
I know you cannot separate the two by genetic, but I thought whether the RNA scope, the resolution is enough that you can see the two dots in each nucleus or only one dot, one cloud for the X's. That was my.
Yep. So good point. So we haven't noticed a second cloud so far, but as I'm sure you know, the resolution of the RNA scope isn't that great. So we're actually going in with higher resolution methods. And the idea is to stain both the X chromosome itself as well as the Xist, so we can really truly co-localize, because I don't feel like we have the power yet to really answer that question with confidence. But it's an important one.
Very quick second question. Yeah. I noticed in the change of genes in neurons, MYC and G2M, which are markers of proliferation. You have proliferating neurons in enough frequency in the hypothalamus that will drive this signature change in aging?
Yeah, so that was a really interesting signature. And again, I don't have functional data supporting this, but I will mention that, so in the case of Alzheimer's disease, as neurons age and they begin to undergo neurodegeneration, a feature of that is that the cells can sometimes try to enter the cell cycle. And so in Alzheimer's disease studies, people have picked up those signatures pretty frequently, as well as the TOR signature, and all related to each other. And so, at least in the case of neurodegeneration, that is thought to be something that kind of puts cells into crisis and actually causes cellular death. I didn't actually expect to pick it up in normal aging, but it may actually suggest that these neurons are sort of primed already in normal aging for that feature. And then what happens is in Alzheimer's disease is something triggers it even further. But I thought those were really interesting signatures as well, and a little bit surprising, but maybe not so much in light of the Alzheimer's disease now that we know. Yeah. All right. So checking the time. Okay, so we have a couple more minutes, so I can answer a couple of questions from the chat.
Yeah, Ashley, if you'd like, I can read them to you.
Sure,
if that helps, or okay.
That'd be great.
So I'll just start with some of the first ones. So this was early in your talk. Reinhard Stoger asked, may have missed this, but was the single cell RNA-seq done on both sexes?
Yeah, so I hope I addressed that, but I will say that we have already obtained the data now on the males. So we've basically redone the whole experiment, comparing directly males and females. We're looking at an intermediate age as well, and we actually have a multiome data that also includes ATAC-seq, because we really want to understand the epigenetic state of the chromosomes alongside the gene expression patterns that change with age. So stay tuned, it's coming, but we're just analyzing those data sets. But I will say that we haven't picked up so far, again, in that data set, Xist did not change in males.
Let's see. So, was there any specific mouse strain that you used in this study?
Yeah, so these were C57 Black 6 mice. So pretty standard strain. It's the most commonly used lab strain in our lab. They're great for things like behavior.
Umet Gerlovic asks, in your first analysis, why did you focus on transcripts only in the nuclei? I think he means for the single cell RNA-seq data instead of the transcripts in the whole cell.
Yeah, so we would love to do whole cell analysis, but the issue with the brain is that the dissociation actually kills most of the neurons. And so we've actually tried it with whole cells in a couple of different brain regions, and you can get really great data back on oligodendrocytes, OPCs, different types of glia, but you just don't get neurons back. And so that's the reason. Although it does come with some limitations, we realize, doing the nuclei.
Next question is, what is the contribution of age-related increase in neuronal polypolarization for Xist expression? And One Ronde says, thank you kindly.
Okay. We have not looked at that. So it's polycomb or polyploid?
Polyploidization. Yeah.
Oh, we haven't seen that so far, but we also have not. We haven't really applied the right tools to directly address that question. But yeah, good question.
So I'm not sure who asked this question, but they ask, have you tried looking at GnRH analogs or antagonists together with exposure to estrogens to see if you could manipulate the mice that way?
Yeah, no, that's a great question. And I think that this study has really raised a lot of possibilities in terms of interventions that we could use related to hormones, related to sex hormones, growth hormone. So I think we have lots of ideas there, but we haven't yet directly addressed any of those questions and how this part of the brain would be altered under different conditions.
I think we might have time for another one or two. Mare Doyle asks, do you know whether Xist is elevated with age in any other tissues?
So I've sort of heard offline just from sort of discussions, informal discussions from other people, but I'm not aware of there are publicly available data out there that if you mine them, you can find it like in these Alzheimer's data sets. But for the most part, I have not seen that published from other groups. I think in part, again, because historically, like our lab as well as other labs, we tend to look at males, right? And there are a number of reasons for that. And I think it's just now that more female data sets are coming out. So I think we'll know soon, but it's not published yet.
Great. Let's see. I'll take one last one.
Mhm.
Robert Clark asks, overexpression of Xist has been associated with affective disorders in females. Is that the same mechanism, you think?
I don't know, but I think that's a really interesting question. And I think that even more broadly, I think that there's a lot to learn from neurodevelopmental disorders, which disproportionately affect males. And the reason for that is that there are a lot of brain-associated genes, or like synaptic genes, for example, on the X chromosome. And males, of course, are more often affected because they're XY, and so you only need one copy of a mutation in order to get the disorder, whereas females tend to be, they're mosaic, and so they may or may not have the disorder. So it's a great, I don't know what the relationship is, and there isn't a lot of sequencing data on those cases right now. So I think that, in the coming years, I hope to get a better handle on that and the relationship between what's going on in our data set and those disorders. I think that's a really interesting thing to think about.
Yeah. So we have a lot of extra questions for you, Ashley, but I think we're going to run out of time. So we'll save those questions either for the roundtable, or if you like, we can address these either. You can type into the